Okay, so let's get started. Okay, so actually this is a small kind of sharing sessions also. So we are going to talk about metagenomics and 16s, particularly how we can get the data from using the nanopore sequencing, and then how we should or how we can analyze or handle our data after we have done the sequencing. So for today, I have prepared some content in terms of some introductions to the backgrounds, the nanopore sequencing, and the technology companies that have the innovations on this nanopore sequencing, which is Oxford Nanopore Technologies, and the technologies that they have at the moment, what is the updates, and also what are the devices that are available. And then we also have two applications. These two applications are only some of the possible ways to look into the metagenomics by using nanopore sequencing, but actually there are much more methods in order to analyze our metagenomic samples and also microbiome samples. So the one that we are going to look into further will be these two, microbial amplicon barcoding and also the rapid metagenomic sequencing. The difference between these two, if we put it into the normal experiments that we have, one is considered a targeted sequencing, is like 16s sequencing or like ITS sequencing or any marker genes, biomarkers types of analysis on our metagenomic samples. Another one is some people will call it as shotgun metagenomics. So we sequence whatever DNA sequence or DNA molecules that we are able to get for our samples. So after some introductions on the background and how the sequencing can be done, and then we will go into the data analysis, particularly by using the Epitome software, which is designed to handle the nanopore sequencing data and also handle kind of from end to end, from the data all the way to the reports, and give us the insights of our experiments and our samples. So introductions to nanopore sequencing. Oxford Nanopore Technologies is a UK-based company, and they develop and sell nanopore sequencing products. So including like all the consumables, all the kits, and also the devices, focusing on the nanopore sequencing. So these technologies has been adopted widely across the world. So up to end of 2024, I think almost 2 years or 1.5 years ago, the number of publications for users who use the Oxford Nanopore has more than 14,000 of them. I think now it should be much more than this number. And then it has been adopted in more than 125 different countries for many different purposes. And then they are different devices to fulfill different requirements or different demands in terms of different domain or different research field. So from the very small throughput all the way to very large throughput. So the visions of Oxford Nanopore is to enable the analysis of anything by anyone, anywhere. So the technologies has been adopted by many different users from different community of different domain experts, and they have been used in many different environments. So including like the fields, the forest, the sea, on the ship. And then I think one of the very special one is they have tested their technologies also in the space. So they have bring the Oxford Nanopore sequencing device all the way to the space station and then do the sequencing over there, and then it works without the gravity. So, and then it also has been applied in many different industries. So for different purpose, including like human genetic research, cancer research, genetic screening, plant and animals, and also one of the very important applications will be metagenomics and microbiome as well. So what are the benefits of Oxford Nanopore? So it can give kind of much richer insights. So in addition to sequencing the DNA molecules like what we have normally either using Sanger sequencing or other NGS like Illumina or PacBio, on top of that, Oxford Nanopore can also sequence directly the native DNA and RNA molecules. So that means we can also get capture the epigenetic information from the sequencing. So like any modified bases in the DNA molecule or modified bases in the RNA molecules, those information is possible to be captured in the Oxford Nanopore sequencing. So in addition to that, it also have the benefits of real-time data streaming and immediate analysis. So for most of the sequencing platform, we need to prepare the samples and then load the samples into the device and then start the sequencing. And we can only see the data and access the data or work on the data after the sequencing is done. This is different in terms of in Oxford Nanopore, because when we prepare the sample and load it into the machine, while the sequencing is still ongoing, we already can see the data coming out from the sequencing process. So it's like roughly 1 hour after starting the sequencing, we can start to see what are the sequence that inside our samples, and then we can start analyzing on it. So this is something like useful, especially for many urgent cases. Also, Oxford Nanopore sequencing devices are actually considered accessible and affordable. So they come with smaller size and also are very portable. So I think I also have one of the sample device over here. So this is one of the device that coming out from Oxford Nanopore Technologies. So common size sequencing device. So this one can be used very easily in many different scenarios. So in office, in lab, in the field, kind of if you want to bring it over to the field, because let's say some of the samples is you need it to be very fresh, and then you can also bring the device all the way to your field settings, and then the sequencing can be done over there, because I think the latest version, they also have the temperature control function. So yeah, it's not necessary to stay in the aircon room in order to do the sequencing. So how does the nanopore sequencing work? So this basically is how the nanopores looks like in the 3D or the structural format. So it's actually a similar, is actually a proteins which has tunnels or channels that inside it. So when we put these nanopores on the membrane, and then we put the DNA molecules which pass through the tunnels of the nanopore, because of the DNA molecules are carrying charges. So when it passes through the tunnels of nanopores, the device of nanopore can detect the current changes or the electrical changes of the pores, and those signals can be converted into the bases. So from there, we know what are the sequence of the molecules that are passing through the nanopore protein. So this is some animations on how it could be happenings in the nanopore sequencing. So the blue color one are basically how the nanopore are placed on membranes or on the flow cells. And then during the library preparations, a motor protein will be attached to the DNA molecule or RNA molecules. The motor protein will bring the DNA molecule or RNA molecule all the way to the nanopore entry, the tunnel entry, and then unzip it and then put kind of let the DNA strand to pass through the pores of it. So when it passing through it, and then the device will record the electrical signals, and then these electrical signals, we will be using some AI models to read those signals and turn them into the base calls. So from there, we will have a single molecule resolution kind of sequence information. So while this happens, actually it happens real time that we can get the signals information in our computer together with our bases or sequences in our computers. So once the data coming out from the sequencing, we can start working on it. We can also do some live analysis. So that means while the sequencing is still ongoing, we already start looking into what's inside our samples. So from this slide, you can also see there's one term, it's called the adaptive sampling. So this is also a very unique features that is provided by Oxford Nanopore. So let me go slightly deeper into this adaptive sampling. Even though this adaptive sampling might not be very useful when we are looking at metagenomics or 16s, but this feature is kind of, to me, is very interesting that I think is worth to share to everyone. So because we know the DNA molecule or RNA molecules will pass through the nanopores, and then we measure the signals of it. So at the same time, we are doing live base calling or live analysis. That means we know whether the nanopores are sequencing the molecules that we want. So in case of targeted sequencing, or in case of we have samples that is mix of maybe hosts and pathogens, and perhaps we do not really need to sequence some portions of the samples, we just need to target certain portion of the samples. Then during the live base calling, we know the nanopores are whether they are sequencing the right DNA molecule or not. So because the base calling happen real time, analysis happen real times, so we can also know which pores are actually sequencing non-target, which pores are sequencing our targets. For those sequencing the DNA molecule that is not within our targets, so we do not really want these molecules, then actually instruction can be sent to the nanopores to eject back or to reject the DNA molecule. So we kind of ask the nanopore to stop sequencing that particular DNA molecule, put it back, and then try to sequence another molecules. So this is some very good functions, especially for cases where you have very complex samples, or you do not really need to sequence everything in your samples. You just want to have a targeted sequencing, yet it's hard to actually use other methods like amplification or prop-based amplification or PCR or primers-based amplification to amplify the targets. Then this is one of the options. So using these methods, we can still also capture the epigenetic information. So the epigenetics, like the modified bases, won't be changed during this adaptive sampling sequencing as well. So it's like very, very useful for many different scenarios, especially those cases we are encountering challenges in sequencing the targets. So nanopore sequencing device, like as shown just now. So this is one of the device, the smallest one. Actually, the sequencing the nanopores or the sequencing are not actually happens inside the device. It's actually happening inside the flow cell. So they are at the moment 2 different flow cells. So both of them, the size will be quite small. So this is the mean ion flow cell. So this is where all the nanopores are put inside. So after having this flow cell, we will have our samples and then prepare into libraries, and then we use pipette to load it into this flow cell. And then this flow cell will be put into the device and then just plug into the computer, and then we can start to do the sequencing. So this is the smaller one. I think the throughput that they have normally is around 10 GB all the way to 20 GB based on our most of the cases. But I think recently our sequencing, some of our users managed to get more than 20, almost 30 GB of data from one flow cell like this. And then this is another type of flow cell. So this one, even though the size look even smaller, but actually the throughput for this flow cell is 10 times compared to the mean ion flow cell. Yeah. So this one normally is used for a bigger project, or if let's say you need a much higher data throughput, then normally this flow cell will be used. And then this flow cell, it comes with another types of device. So depends on the flow cells that is used in an experiments. So we have different devices from ONT, which can help us to handle the sequencing. So for mean ion, at the moment, there are 2 different devices available. So one is the mean ion MK1D. So this is the latest model. Another one is grid ion. So mean ion, as you can see from the size, at one time it can help to handle one flow cell. So it's like one sequencing, one flow cell, and then roughly you should get 10 to 20 GB of data. And then the grid ion is actually having the capacity to handle 5 flow cells at the same time. And it comes with the GPU, so that means you do not really need to have a very high power computer in order to do the sequencing or handle all 5 flow cells. So it comes with the GPU card, and then it can help to do the base calling real time using the device. If you are using PromiIon flow cell or requiring higher throughput, then actually at the moment there are 3 different types of devices available. So PromiIon Solo, PromiIon 2 integrated, and also PromiIon 24. So from the name, actually, we can also guess how many flow cells it can handle at one time. So both the PromiIon 2 Solo and PromiIon 2 integrated can handle 2 flow cells at the same time. The difference between the Solo and the integrated is one is do not contain any GPU or processing power. Another one contains the GPU, and then it can help to handle the library calling directly without depending on the computers. And then, of course, the higher throughput one will be the PromiIon 24. It can handle 24 flow cell sequencing at the same time. So these are the devices that are available at the moment. So from the sequencing, actually what we can do for our projects or for our samples. So one of the workflows that is tested or developed by ONT is called the MAB, which stands for the microbial amplicon barcoding. So one of the very kind of widely studied topics will be the metagenomics, and then we normally would like to know what are the bacteria, archaea, or even together with the fungi, community members in the samples. So by using this particular workflow, we can supposedly to identify the prokaryotes through the targeting the 16s ribosomal RNA genes, and then by using the primers or the amplification for the ITS sequence, then we can identify the fungal members in our samples. So this particular workflows, how it works basically, starting from the genomics, gDNA, there are pre-designed 16S or ITS or both primer set that can be used for the experiments. And then by using these primers, the amplicons of the targeted regions will be amplified and then put into the libraries. And then with the libraries, we can load into the flow cell and then further put into the device for sequencing. So how long it takes to do an experiment such as this one? So this targeted metagenomics workflow developed by ONT, the sample extraction or how to get the genomic DNA out from the samples, that one will be depending on the users, how long they need to do the DNA extraction. And then once the genomic DNA is ready, then preparing the library or estimating, we are estimating around 3 hours, and then the sequencing, it can take times 12 hours all the way to 72 hours, depending the number of samples that you have or how much throughput that you would like to get for each of the samples. So because the sequencing happens in real time, so while sequencing is still ongoing, you can observe or monitor the progress of the sequencing. So when maybe up to few hours, you already see you have sufficient data for your samples, then you can stop the sequencing and then perhaps watch the flow cell and then reuse the flow cell for other experiment. And then if let's say you have a lot of samples multiplexed into one sequencing run, and then you need to have very high coverage for all the samples, then perhaps you can let the sequencing run all the way to up to 72 hours. So and then after the sequencing or during the sequencing is running, we can do the analysis, and then the analysis can be handled by another program called Epitome. So later we will go deeper into this Epitome software. And yeah, so this is basically what we are seeing. Maybe after you have the DNA extracted, and then maybe half day to prepare the library and then to start the sequencing. And then you can let it to run, or perhaps you can come back before you go back to home, or you come back the next morning to see how's the progress of it. And then if sufficient, we can stop the sequencing anytime. So it's like, and then the analysis, normally it could be as fast as within 1 hour. So for one experiment, it could be as fast as you start everything in the morning and then get the results before you go back home and call it a day. So this is how the experiment could happen. And then for this 16S or MAB workflows, actually is targeting the full-length 16S genes. So compared to other applications using other platforms such as Illumina most of the time, because of the restrictions in terms of the read length, we normally able to sequence only partial 16S genes. So perhaps the V3 to V5 regions, if let's say you are able to get the sequencing length more than maybe 350 or up to 500 base pair in total forward and reverse reads, then normally we are able to sequence perhaps V3 to V5. But if let's say normal platforms, it could maybe up to V3 to V4. And then if we want to identify certain class or certain families of bacteria, maybe we need to change our target to V9 or other regions of the ribosomal genes, because the technologies might not have the capacity to or capability to sequence the whole gene of it. But for Oxford Nanopore, it's actually to basically the maximum DNA molecule that it can sequence is up to MB in size. So there's definitely no problem to sequence the whole genes of 1.5 kb for these 16S genes. So these are the primer sequence, and then this has basically are the improved versions of the primer design. So with these primers, from one sequencing run, we can capture actually very comprehensive metagenomics information from our samples. So these are the benchmark against using the zymotic Bioinformatics microbial community standards. So they have tested it, and then basically it works, and then it should be no PCR bias or target region bias. So you might, because let's say the bias happens, then you might see some of the bacteria seems like being overrepresented in the samples, and then some samples are underrepresented. So this one, this workflow supposedly to have less bias, and then you should be having a clearer pictures of what are there inside your samples. So another possible ways to identify the members inside your samples would be these rapid identifications. Basically, it's not restricted to the respiratory pathogens, but it should be able to kind of work on many different types of samples. So these methods basically is suitable in the case that if you want to identify the potential pathogens or any potential members inside your sample, but you do not really know what could it be. So perhaps in a scenario like an outbreak or maybe some infections happenings, and then you would like to know what could be the pathogens. Maybe it's a virus, maybe it's a bacteria, or it could also be a fungus. So in that scenario, we just sequence whatever DNA sequence inside the samples and then try to identify what could be the cause of certain pathogen, certain infections or certain outbreaks. So these workflows can quickly identify the pathogens from, they are using the respiratory samples as example because I think they have tested quite a lot since the SARS-CoV or the COVID era. So this is how they recommend to prepare the samples. So because I think most of the time there are some complications in order to identify any unknown virus inside of samples, because the virus could be RNA virus, it could also be DNA virus. DNA virus, it could be single-strand DNA virus, or it could be double-stranded DNA virus. So that's why they have some recommendations in order to captures whatever virus inside the samples. And then for bacteria and fungus, basically we should be no problem to capture the double-stranded DNA inside the samples. But normally the samples could be contaminated or is mixed with the host DNA. So let's say the samples obtained from human or from clinical settings, likely the DNA samples that we have contain a lot of human DNA. So we might proceed to do some host depletion, for example, like using saponin, so that our samples are enriched with whatever non-human DNA molecules. So this, how long it takes, actually is quite similar to the MAB. So starting from the genomic DNA extracted from the samples, and then it takes roughly a few hours to prepare the libraries. And then after the library is prepared, I think less than half day, we should be able to get the libraries done and then load into the sequencing device, and then let the sequencing happens. So perhaps we just let it run, and then before we go back home, we should be able to see some of the results from the sequencing run already. So it's like some urgent cases where you might get the samples in the morning and then extract the total DNA, total RNA or total samples from your case, and then by the end of the day, you might already have some idea what is going on with the samples. So for this protocol or for this workflow, they have also done the testing. So for more information like what is the detection limit or what is the sensitivity of this workflow, you can try to look into their testing or the assessment results over here. So this one, they are using some respiratory panels to do the testing, which include all the virus and some bacteria and even some fungus as well. And then they try to see how much how is the sensitivity in terms of these workflows. So whether we can detect the bacteria or the virus pathogens if, let's say, the titer of it is very low in our samples. So after the sequencing or during the sequencing is happening, we will get the data in our computers. So from there, it could be quite challenging for some of the users, especially biologists, because the data might look complicated, and then we open source programs or any fast programs, they might come in command line. So that's why Oxford Nanopore also have provided solutions for everyone to work on their nanopore sequencing data. So this actually for this webinar, we are going to focus more on this analysis part. So once the sequencing is done, how we can handle our data. So for most of the time, we have two types of bioinformatics tools to help us analyze on the data. So one is a easy one with graphical user interface, and then normally how we use it is just point and clicks. Another one considered more challenging for most of the people will be the command line. So for nanopore sequencing data, actually it can be handled by software that comes in the set of graphical user interface, or it can also bioinformatician can also analyze them by using some command line tools. From ONT, they have developed two major software or two main software. One is called the MinKnow. MinKnow software is instrument control device, instrument control software. So it can help us to start the sequencing, to monitor the sequencing, or to handle the sequencing parts. And it also have it carries some analysis functions. So in case you need some fast analysis directly from the sequencing, MinKnow can also help you with that. But if let's say you are looking at a more complicated or more in-depth kind of analysis, then you might consider Epi2Me software. So this Epi2Me software also a point-and-click software, and then they come with pre-configured workflows or pipelines. So you do not need to worry about what are the steps that you need to carry out for the different settings. And then, of course, if let's say you still prefer to use the command lines, the Epi2Me or the pre-configured workflows can be run also through command line. And then over the years, there are a lot of users from kind of using the ONT technologies to do their sequencing and experiments. And then they have also developed a lot of tools to handle the nanopore data. So if you are looking there are some software or programs that are suitable for your case, you can also use them for handle your nanopore data. Just that the pipelines that are built from the community might be supported directly by the developers themselves. And ONT will be supporting all the software or workflows that developed by them and shared to the users by them. So the pipelines or the workflows includes the analysis for metagenomics and 16s. So to help identify the members inside the samples, it can also contains the workflows that helps with identifying the human variations under the whole genome sequencing of human samples. And then if let's say you are looking into the transcriptome or RNA sequencing at the single cell level, there are also pipelines that helps you with that. And also biopharma QC, let's say you are doing a lot of plasmid, and then you want to check your plasmid sequence, whether they carry the right genes, whether the inserts are correct, and etc. That can also be done, you sequence the plasmid, and then there are analysis workflows to help you confirm all the genes or all the content inside the plasmids. So let me go deeper into this Epi2Me. So this EpiTree is a desktop application, so we download in our computer and then use it in our computer. So it won't have the problems like you don't need to upload the data to somewhere in order to run the analysis. And then it's free. And then it comes with at least more than 15 different pipelines or workflows. And then the setup is quite straightforward. And then it can be installed in different operating systems such as Windows, Mac computer, Linux. So yeah, it's quite flexible. And then so far from what I've tested for 1 year plus, it's still running very smoothly. And then all the updates is kind of very easily can be done. And then switching different versions of workflows, all that can be done quite easily without requiring to look into the codes or look into the commands. So how it works, so our files, because after the sequencing, supposedly it should be inside our computer, or in case you are not really doing the sequencing on your own, and then you get the data from your lab mates, your collaborators, or perhaps you also might have data that come from a public database, you get it from some publications, and then you download the data. All this can be done, the analysis can be done by using the EpiTree as well, not a problem, as long as you have the data with you in the computer, and then you have this EpiTree software installed in your computer. So inside this software, actually there are options to choose where you want to run the analysis. And then by default, the analysis will be carried out in your own computer. So all the process, all the tasks is run totally locally in the computer. But some case, we might have computer that is not powerful enough to help us with the analysis. And for this, actually, ONT is very generous in sharing their cloud resource. So in case your computer is not strong enough or not high spec enough to run certain pipelines or certain analysis, you can actually also use the cloud resources provided by Oxford Nanopore. And then for the cloud, you do not need to identify where you need to register the cloud or how you want to start the cloud and then where you want to upload your data. So everything is covered within the EpiTree software. So just switch or kind of select where you want to run the analysis within the EpiTree software, and then that can be done very easily. So the pipelines available in EpiTree at the moment, for human genetics, there are 3 different pipelines available at the moment. So trio analysis, human variations, and also somatic variation. So if you're looking at some maybe rare disease cases where you have the samples from the proband together with the parents, then this workflow is something that is useful for you. And then you have maybe human related or human sample sequencing, whether it's targeted human sample sequencing or whole genome sequencing. The human variations pipelines will be useful. And then in case you have cancer samples, you have the pair cancer samples together with the normal sample, and then you want to identify the somatic variation, then somatic variation will be good. And then some basic tasks like alignment and base calling. And then, of course, metagenomics and 16s, there are 2 pipelines for that. Quality control, let's say plasmid sequencing, there are pipelines specifically for that purpose. Transcriptome sequencing, general bulk RNA-seq, or if let's say you have single cell RNA-seq. Targeted amplicon sequencing, and then for infectious disease, bacterial genomes, and then these particular pipelines now also can be used for fungal genome sequencing and assembly. So there are actually several pipelines available for other types of pathogens as well. Some might not be listed here, and some might have they have obsoleted. One of the very commonly used pipeline will be the one that we use for the SARS-CoV-2 genome sequencing. And then for genome structure, so if let's say you're aiming to get a very big genome and then using long-read sequencing or nanopore sequencing, on top of that, you can also further improve on the big genomes, so using the Talosic and also the PORC. So there are also pipelines to help you out with that. So this is some further information on the EpiTree workflow. So in case you still prefer to run it using command line, The workflows are also shared publicly in GitHub, so you can also try to look at the code of it if you, let's say, you are interested in that. So for this demonstration, I will be going through to show you how the data, the nanopore sequencing data looks like, especially after a sequencing or during the sequencing is running, and how we can get the Epi2Me software downloaded to our computer, and then how we can install it in our computer. And then we will go deeper into two analysis pipelines or workflows. One is designed for 16s or amplicons. Another one is for general metagenomics analysis. And actually, there are some further functions that we can kind of play around with these Epi2Me workflows as well. So I'm going to show you with that later. So with this, let me try to pull out my browser. So, okay. So where to get the Epi2Me? Epi2Me now basically is open for everyone to download and use. So where you can get it from this site. So maybe let me put it into the chat box for you. By the way, in case you have any questions regarding the information for this webinar or anything that you would like to ask, feel free to drop it into the chat box. We will be looking into that shortly after the demonstration is done. So this is where we can get the Epi2Me software. So if you scroll slightly down, there's a download button over here. When you click over that, you will see different installer for the different operating system. So depends on the operating system you have, you can get the right installer over here. And then actually at the bottom of this page, there's also installation guide. So different operating system will have a different installation methods. So when you click into this page, it will show you step by step, very details on how to install the Epi2Me software. It's actually complicated, considered slightly complicated when you want to install it in Windows, because this particular software make use of the Windows subsystem Linux. So it will, during the installation, the Epi2Me installer will need to get some permission from you in order to enable certain advanced function in the Windows. So how the steps will go, all the screenshots are shown over here. So supposedly there should be quite straightforward. So if you, without looking at this page, you follow all the next, and then allow it to finish up all the setup, then supposedly it should end without any problems. But from my experience, some of the steps, especially when the WSL is activated in the Windows, some computer restart will be required. So I wouldn't suggest you to try installing right now. So you can try it while you have nothing else running in the computer, and then because during the computer restart, some of the ongoing activities might be stopped during the restart. So for Mac OS, there are also some guides on how to do the installation. And then for Linux, same thing. And then, yeah, only the Windows might need to go through some complicated process. But all these process are basically handled by the Epi2Me installer. So for users, we normally just need to follow through the process. So one is install. Then let me show you how it looks. So after the installation is done, I think the first page that you might be seeing once you open up the software is asking you to sign in. And this one, you can sign up a free account for ONT. So once you sign up using directly inside this Epi2Me software, you can sign in, and then because the sign in is required if, let's say, you are using the cloud resource. But of course, if let's say you still prefer everything run in your computer and you do not really want to run it using cloud, so perhaps you still can able to log in as a guest without any login. And then once it's logged in, and then all the account, the account settings will be here. So you can try to see whether you are logging into the right accounts. And then once the login is done or sign in as guest, then you will see the dashboard like this one, which shows some general status of your Epi2Me session or your computer status. And then if let's say there's any new updates for the software available, then it will also show a notification over here. So, and if want to update the software, we can proceed to take the details and then complete the software update accordingly. And there are also some news from ONT which will be shown over here. So yeah, when they update their software, they will also tell us what are the latest updates that they have implemented in the later version. So you can also try to look into their blogs. Most of the time, they are very updated in sharing all the information that they have in ONT. And then for users who install this software for the first time, normally I will also suggest to take a look at the settings at the lower left corners over here. So once you click into these settings, and then you will be seeing where is the working directory of it. So this is where all the analysis is carried out. So all the files will be basically keep in these particular positions. So if you want to switch to another, maybe other hard disk or etc., you can do it over here. Or else we can leave it as default by using the user account, and then it will create a folder called Epi2Me Labs over there. And the things that normally I want to kind of emphasize more will be on the local tab over here. So this one can help us to check whether the software is ready to run analysis, because most of the pipelines, it required the dependency. So one is it need to enable the Windows subsystem Linux. So if let's say it's already activated, then you will see the status is ready to be used. Another dependency or required software will be the next row. So if let's say these two software are not available or not yet available, we can open setup over here and then ask Epi2Me to help us finish up all the setup for these two particular programs. So once you see the green ticks is ready, and then these two particular programs, when is not active or not being used for a certain time, it will go into sleep mode. So sometimes we also might need to come back here in order to wake it up. So if let's say you see you have done the setup, but sometimes when you come back, this page is showing that it's not ready in red color or maybe yellow color is checking. We can also click the open setup over here and then click the retry to refresh it or to wake it up. So one is in green ticks, normally it's no problem. And then the system resources, you can set it over here. So that means how much computer resource that you want to allocate for this Epi2Me while doing the analysis. So how much threads or cores, and then how much RAM you want to allocate. I won't recommend to use up all the RAM particularly, because Windows, when normal use, we are going to need some RAM as well. So leave at least maybe 8 GB of RAM for your normal Windows activities, and then for analysis wise, perhaps you can use whatever remaining. But if let's say your computer is like my personal computer, perhaps another computer with 16 GB of RAM, then perhaps we can use maybe all the way to 15 GB of it, and then leave roughly 1 GB, and then not really running anything else in the Windows while the analysis is carried out. So the setting, when it's done, we just need to, we actually have only 3 buttons over here. So the home basically is go back to the dashboard that we see just now. And then other 2 buttons, one is launch and the other one is results. So launch basically is when we want to run certain analysis, and then results is when we want to see the analysis results. So when we click the launch, and then supposedly you should see the pre-configured workflows available in the Epi2Me. By default, initially it won't be installing all the workflows for you. So you can check the lower right corner for each workflows. If you see the icons of computers being light up, then that means the workflows have been installed in the computer. If no, then if you want to use it, we need to do some further installation. For example, like perhaps this one. So the policy, you can see the icon is gray out. So that means this particular workflow is not yet available in my Epi2Me. So if I want to use this workflow, I just need to click into this one and then click the install button. and Epitomy will settle everything for me. So after installation, then we should be seeing the computer icon of that workflow being light up. So this one, when we click install, it will also suggest what are the computer requirements that we should prepare for this workflow. So what is the minimum and then what is the recommended. So in case the computer system might not have sufficient CPU or RAM, we still can able to install the program, and then because after installation of the workflows, let's try to use this one as examples. All the documentations will be inside here. So for example, for this metagenomics pipelines, it comes with the manuals on how to use it and some description on what is this workflows about directly in Epitomy, so you do not need to go to website or ask ChatGPT or what is this workflows about. So the most precise and accurate information is supposedly to be here together with this workflow. So inside this manual, most of the time it starts with introductions and then what are the general overview of it. And then importantly, it also shows you what is the input requirements, what are the outputs will be coming out from these pipelines. And then for me, normally I will also look into the pipelines details, like what are the steps that will be carried out, and then what are the software that it will use to carry out the task for each of the steps. So this one, normally it will have many details on each workflows. And then in case we run into problems, then we can further look into the troubleshooting guide and also the frequently asked questions over here in these workflows. So other than this documentation that comes with the workflow, on the right side, you can see the control panels like this one. So if we want to run the analysis in our computer, we just leave it as run locally. And then if we want to try running the analysis using the cloud provided by ONT, we can switch it to run in cloud. So same, it looks very similar, and then because basically it's running the same analysis or same analysis pipeline, but in different places only. So back to this run in local. From here, you can, when we click the launch is when we want to start the analysis. So before we start the analysis, we can try to look into some additional information here. So options, normally first time after the workflows is installed in Epitomy, I will run one round of demo analysis just to make sure my computer can finish the pipelines without any problem. And also, if let's say I have not get my data yet, then I can run the demo analysis to see what are the expected input and output from these workflows, and then what are the how the reports looks like using this workflow. And then from time to time, I can also check the updates. And then if let's say I want to switch to different versions, I can do it over here. If I will no longer require the workflows, I can delete it from here. And then if I want to see the details, I can open the folder of it and also install some optional dependency if required. And then the versions that I have right now is this version. Yeah. And then if I want to start the analysis, very easily just click the launch button over here. So for metagenomics analysis, from the sequencing run, normally we will have the data. So let me quickly show you how the sequencing data looks like. So after a sequencing is done, if let's say I'm using this computer to do the sequencing, normally we will have a folder called data, and then inside the data folder, we will have one folder for each sequencing run. And then let me take this as example. So this is one of the sequencing that was run last year. So when the sequencing is done, we will have a folder like this, and then inside the folder all the way to several level down, we will have the sequencing reports and sequencing log files and details like this one. And then part 5 is normally the very raw electrical signals files. So it's not actually analysis ready. The analysis ready files normally in FASTQ format or in BAM file format. So FASTQ, inside the FASTQ pass folder, if we have multiplexed many samples into one sequencing run, it will have one folder for each barcodes or for each samples. And then inside each samples, we will have multiple FASTQ file. This is because we have done live-based calling. So every, this one, I think previously every 10 minutes, we are going to get one set of data. So that's why the data keep on, it collect all the data from time to time into these folders. So one sample will have multiple FASTQ files. So this is how the data looks like. And then in case you get the data from your collaborators or you get it from public database, most of the time it's also in the FASTQ format. And sometimes it could be in the BAM format, especially if, let's say, you are also looking into the modified base information. So back to this Epi2Me. So you can see the setup page over here. All the parameters have been pre-configured or selected for you. So what we need to do is select what is our input data, our files. So we have two options over here. So if, let's say, we get our data from perhaps public database, most of the time we will have one FASTQ file for one sample. So in that case, we can select, let's say we just want to analyze one sample, we can do it by selecting one file for that case. But because like what you have seen just now, the data directly from nanopore sequencing, we will have a folders containing multiple folders, each folder for one sample, and then each samples have multiple FASTQ files. So in this case, normally we can directly select the folder as our input and then select the FASTQ pass folder as our input folder. So this one is actually the 16s data, but it's okay also because the metagenomics data pipelines, it can take in many different kind of data as well. So inside this FASTQ pass, for this particular demo data set, I have 3 samples, and then each samples is carrying different barcodes. So like this one, so I can select the whole FASTQ pass folder as my input. And then if I do not really have anything else to tweak in my pipelines, any parameters, I am okay with all the default ones, then straight away we can start the workflows. So just to further explain a bit, because this one metagenomics, for this particular workflows, there are 2 different classification methods or taxonomic profiling methods. So one is using the Kraken tool, another one is using the Minimap tool. So 2 different approaches. Kraken tool is a faster approach, so it can tell you what is inside your sample in a very short time. And Minimap takes longer time, but the result is going to be more robust and it's going to be more accurate. So depending on your requirements, if you want to know the result fast, you can try the Kraken tool. And then if you want to have a more accurate results, you can try Minimap tool. And for my case, most of the time, I will run both. Supposedly the result from both analysis methods will come into consensus. And then for reporting, perhaps I will be sticking to the Minimap tool. So other than the classification methods, by default, the sample names will be based on the folder name that we see just now. So sample 1 will be barcode 01, sample 2 will be barcode 02, and etc. So if you prefer to name your sample accordingly instead of barcode 01, barcode 02, you can also provide a CSV file or a table file so that it can know what is barcode 01 and what is the name that you want to give to barcode 01 and etc. So it can be set over here. And one important information or parameters that I would like also to highlight will be this reference. By default, it's going to search our sequences, our samples against a database called Standard 8. So other than this Standard 8 database, there are also other database that is prepared for Epi2Me to use, including like the 16s and 18s sequences from NCBI, and then a more comprehensive one together with the 28s and ITS. And some user might be looking into 16s, prefer to have a more curated database like Silva and Green Genes. So all these databases have been pre-compiled into this Epi2Me, so you do not need to prepare the database on your own. And then if, let's say, you still prefer to use the Kraken database, there are also options for Standard 8 plus PF and plus PFP. I will come back to explain more on these 3 databases, what are those databases shortly. And if, let's say, you have your own Kraken database that you would like to use and then it's not available publicly, then you can also get it kind of prepare and then look into the Epi2Me to do the taxonomic classification accordingly. And also by using Minimap 2, you can also provide your database or your reference in the FASTA format and then link the sequence to the taxonomy ID accordingly. So, and then by default, the taxonomic rank, the pipeline you try to identify is up to the species level. So of course, feel free to adjust the taxonomy rank that you want to achieve over here. Just one thing to note is some of the curated database, such as the Silva database, even we can set the taxonomy rank up to species level, but the database only carry information up to genus level. So the results that we will be seeing, even though we select species here for Silva database, we still be seeing only up to the genus level. So further parameters tweaking over here for Kraken2 and Minimap2. So optimizations can be done if, let's say, it's required. And then for samples, shotgun metagenomics, and then if you, in case you also want to check whether the samples contain any antimicrobial resistant genes, you can also enable this part. And output, like what are the additional outputs that you would like to get from the pipeline is? And what is the any other cutoff that you would like to set to the samples or data before the analysis is done on it? Some next flow configuration. So this one, every analysis runs inside this Epitome will assign a name to it. So this name is random, two words that is random. So if you want to use a more readable names, or we can directly change the name over here. So for example, like this is a demo meta run or something. So whatever that is meaningful for users, we can do it over here. So yeah, so when all the ticks are in green color, that means the pipeline is ready. So we can click to launch and run it. So it might take a while to check whether all the requirements are ready for the analysis or not. For users who first run this pipeline, it could take slightly longer time because it need to download the database accordingly. So the one that we are seeing just now, we are using standard 8 database. So for the first time, you need to download the standard 8 database. And then for the subsequent runs, using the same database, as long as the database has been downloaded in any previous runs, then we don't need to redo that step. So the running time will be much faster. So once the run is kind of activated or kind of started, then we will come to this particular run page, which show what are the steps that are being carried out by the pipelines. And then if, let's say, we see all the green bars, then that means the tasks have been carried out successfully without any problems. So we will come back to this later. So normally the results can be, we can check the analysis run from here. So this one, it could take perhaps few minutes in order to analyze the 3 samples that we have put in. So while waiting, maybe I can show you sorry, where is my browser again? Yeah, this one. So just now we have mentioned about the databases. So what are those standard 8, PF8 and other database? So actually they are Kraken2 databases, and then they are prepared by the developer of this Kraken2 software. So, and then what is standard 8? Standard 8 database basically is a standard database capped at 8 GB in size. So standard database, basically they are RefSeq, archaea, bacterial, virus, plasmid, human, UniVac sequences. So the full database is in the size of around 96.8 GB. It's very big. So the standard 8 is actually a small subset of 8. So for Epitome, the default one is going to use a small subset, but in case you prefer to use a more comprehensive, a more robust database, let's say 16 GB or all the way to the full size of it, we can download the Kraken database from this site. So this one is also prepared or kind of supplied by the Kraken to the team. So this one, in addition to the standard database, there are also plus PF and also plus PFP. So plus PF basically is the standard database, and then they add in the protozoa and fungi sequence. And then plus PFD, the protozoa, fungus, and also the plant sequence. So it depends on your sample condition or your experiments. So if you think your samples would have also the protozoa, fungus, and also plant sequences, then we will be using this plus PFP. Plus PFP, the full size of it is more than 200 GB. So it's very big. And then optionally, we can also use the subset of it like 8 GB or 16 GB of it. And then there are also other database available over here. So like the virus database and also some GTDB database come with different sizes. The estimated sizes are all listed here. And then it also have some specialized database such as the one designed for 16S like Green Genes and Silva, and also some of the database from time to time. So this one I think is quite useful for many users like us to check what are the suitable database for us to use. And then once the database is downloaded in our computer, we can also load into Epitome to use. So where to load it, and we can come back to these workflows again. So under the reference option, if let's say we are going to use the one that we downloaded on our own, we can select the .tar.gz file downloaded directly into in these particular options, and then the analysis will be carried out doing the taxonomic classification by using the database over here. And one thing to note that is for the database that we are seeing just now, according to the developers, when we are going to use a data set that is roughly 8 GB in size, our computer need to have around 8 GB of RAM or memory, because the first step of the analysis is they try to load the whole database into the memory or the RAM of the computer before doing the taxonomic classification. So in other way of saying will be, if let's say we want to analyze a database with around 200 plus GB, our computer should have the memory around more than 200 GB in size. So that's why it's actually quite challenging if you want to use a very big database such as this one. Okay, so other than that, let me go back to Epitome software. So we also have a specialized pipelines for 16S. And these particular pipelines, actually, they are running the same pipelines compared to the metagenomics that we are seeing just now. So it also have both the methods of Kraken 2 and Minimap 2. The difference is this pipeline, they have optimized the parameters for 16S or any amplicons. So for 16S, because the read size that we are getting most of the time is going to be 1.5 KB, so 1,500 base pair. So it actually comes with the cutoff of 800 base pair as the minimum cutoff and maximum 2,000 bases as the maximum cutoff. So some of the cutoff on the selections in terms of parameters over here is optimized for 16S. So it's up to user. If you prefer to do all the optimization on your own, you can try to use the metagenomics, which is more general in terms of analyzing the data. And then if you have used the MAB or any specialized workflows by ONT, you can directly use the 16s rRNA workflows over here, which is more designed or more optimized for the 16s data. So other than that, maybe I can show you also how the results looks like. Yeah, I think this one I just ran a few days ago. So this one I run the 16S using the Silva database. So this is how the reports looks like. So after the analysis is done, it will have a report over here in the run page also. So from here, we can see what is the general data QC over here. So for barcode 0, 1, 2, and 3, So how's the quality looks like, and also what is the read length we obtain from our data. So this is 16s data, so we can see very obviously the read length is almost at the size of 1.5 KB. And then what is the data throughput for our data, and etc. So for different samples, we can switch to view them over here. And what are the number of reads that we obtain for each samples, and then how many percent or how many reads are actually cannot be matched to the database that we have selected. So this one I think is very good because most of the sequence come from 16s, and then we have actually very low backgrounds in terms of the sequence. So here it have the kind of the visualization on what is the members or how is the compositions of the organisms or bacteria in our samples. So by default it's actually showing the order. So of course we can switch to other taxonomy rank, because this is SIVA database. It only have information up to genus. So even though we select species, it's going to show the information all the way maximum up to genus level. Of course, we can also switch to look at different samples over here. And the sunburst plot, or some people may call it the corona plot. So to see the compositions and then further investigate. Let's say I would like to know more about the formicutes, and I just need to double click to check, to zoom into it, and then refresh to restore it. And then across all the samples, what is the compositions? And by default, it's showing the phylum over here. We can also switch all the way to the genus level to see whether there's any difference in terms of the compositions for our samples. And then for metagenomics, I think one of the very important metrics for users to look at, basically this, by the way, this is the read count or the abundance table for each of the taxon that we have observed. So of course, this one we still can switch to the taxonomic rank that we want to look at. And metagenomics, many people are looking at the diversities of it. So diversity has also been computed by using all these alpha diversity matrices. You can kind of refer or download these tables for further usage as well. And then whether the sequencing is sufficient for our samples, normally we look at the richness curve. So if we are seeing the samples becoming plateau over here, that means the sequencing should be sufficient for our samples. And then if let's say it's keep on increasing, maybe we don't, if the sequencing is still ongoing, then we will continue to let it run until we see some plateaus for the samples that we are interested in. And then the distribution of the abundance by samples can also be shown over here. So yeah, it's a very comprehensive reports together with the software that is used together with the version of it, and then the parameters of it. And what about the results files? So all the analysis results file can be found over here in the file tabs. And then if let's say alignment is carried out, and then we want to see the alignments, a light version of IGV is also incorporated into the epitome. So for us to view the alignments directly from epitome here. And then the last tab is a log tab. So most of the time, we don't need to look into this particular tab. But in case this analysis run into errors, we will be looking into this log in order to identify the problems and then try to troubleshoot from here. So this one, if let's say there's any errors happening, the error message will be shown over here. And then one way to troubleshoot is we can copy and paste the whole error message to any AI chatbot like ChatGPT or Gemini to help us troubleshoot or suggest what we can do. And alternatively, if let's say you need some further help on that, you can also reach out to NextGen, or let me show you some the one that actually run into problems. Yeah, so it could be something like this. If let's say it runs into errors. So yeah, some of the steps might not be able to complete like this. So from the logs, we can try to see what is the problems that come with the error. And then we can copy the whole message from here to ChatGPT to ask what we can do to rectify the problem. Else, we can also click on the report issue on the upper right corner. And then this one, after the export, it will create one zip file. And then you can email the support at nanoporetech.com to ask help from ONT directly. you can also perhaps reach out to NextGene, and then let's see what we can do to help you with the errors, or what are the things that we can identify from these files in order to kind of rectify the problems or solve the problems. So, and then for all the runs that is completed, yeah, this is the one that we run just now, and then it's now completed, and then the reports is over here. And then one of the things that normally I will also show to you will be, if let's say we want to change the name, it can be done over here. And then output files, reports files, we can also look into it from here. So it's going to be have all the files inside our computer in this particular directory inside the output. And then there should also be a reports in the HTML format. This HTML, basically you can share to anyone, or your collaborators, your friends, your lab mates, your supervisors, for them to also take a look at the analysis results. So your friends or your collaborators, they do not need to install the Epi2Me in order to look at the results, because this reports is in HTML. So they just need to have the web browser in order to open up the report like this. So it's fully interactive, like what we have seen in Epi2Me as well. So with this, basically, I think I have demo to you most of the functions or features on how to use, or how we can use this Epi2Me to help out with our experiments or projects. And let me see, anything else I should be showing to you. Oh, okay. So in case the analysis, we do not really need it anymore, we can also delete it from here. So we can take it from here and then delete the instance, or we can also go into the analysis. Once confirmed everything is no longer required, we can also delete everything from here. So in this case, once the analysis is deleted, we do not need to find back where are the files were stored previously in the computer, because this one will help us clean up and then free up the storage space. So yeah. So with this, basically, I have done the demonstrations on the metagenomics, and then for 16s, supposedly to be the same ways on running. And then the database or the references are very similar as well, except for you don't really put in the Kraken's database over here, because Kraken database is a more general database, but not a 16s database. Okay, so it's quite easy to use this particular Epi2Me software. Very user-friendly, very intuitive. So yeah, I have already showed you the Epi2Me, where to download it, how it can run the analysis by using Epi2Me. And then in terms of custom database, where we can get the custom database and prepare our own database as well. Yeah, for this session, we have gone through briefly on the Oxford Nanopore technology and the nanopore sequencing from them. And also we have gone through some how we can design the metagenomics related workflows, and also Epi2Me programs, and how we can run the epigenetics or metagenomics analysis. And before we end the sessions and go into the Q&A, so just to further share a little bit on, we have some upcoming events on the ONT as well. So soon, I think by next month, 16 June, we will have a symposium carried out in Sheraton Hotel, PJ. So this one, we have invited many ONT users, and then to share their experience or their projects using the nanopore technologies in their experiments or their projects. So this symposium will be more focusing on the clinical decisions making. So we will be seeing a lot of users from the clinical settings. This is a physical event, and then it comes with some minor registration fee. And then, of course, the one that you are joining right now is the Bioinformatics webinar series. So it's free and online. So if you're coming back next week for Amplicon nanopore sequencing data, then we will see you again. And then subsequently, we also have other sessions for different applications as well. So feel free to share to your friends or colleagues as well. in case this is going to be helpful for them. So with this, thank you so much for your time staying with us all the time. I think I have overrun around 15 minutes. Yeah, sorry for running over the time.
No worries,
So,
thank you for the very informative sharing. So now we would like to open the floor to Q&A section. Anyone want to raise the questions? Please feel free to do so. And we will be very happy to discuss with you. Anyone?
I think so far I don't see any questions. So I think likely we are going to leave the session continue to run for another 10 minutes while we are kind of turning off our mic and also my presentation screen. But you still can drop in your questions to the chat box after this, or reach out directly to NextGen in case you need some further information from us or you need some help from us. Oh yeah, Luis, how about the feedback form?
Yeah, before we ending this, I'm just sent here the feedback form. So everyone please help us to take about 5 minutes to click on the link and fill it. And the e-certificate will also be distributed after the completion for this form.
All right, thank you so much for everyone's time. So we'll see you again.
Thank you everyone for your time and support. So we hope this session will provide valuable insights for your support, research, and future projects. And look forward to see you in next webinar. Thank you so much.